Thank you everyone for joining us, and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Thursday, June 25th, 2015, and the title for today's webinar is Getting to Cures, the Next 5 Years in Immunotherapy. In this webinar for patients and caregivers, we will discuss the current advances in cancer immunotherapy and where we see us going in the next 5 years. Today's webinar is made possible through the generous support of Amgen. Amgen focuses on areas of high unmet medical need and leverages its biologics manufacturing expertise to strive for solutions that improve health outcomes and dramatically improve people's lives. A biotechnology pioneer since 1980, Amgen has grown to be one of the world's leading independent biotechnology companies. It has reached millions of patients around the world and is developing a pipeline of medicines with breakaway potential. We thank you, Amgen, for your support. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. Cancer Research Institute is the world's only nonprofit organization devoted exclusively to harnessing the power of the immune system to conquer all forms of cancer. We fund scientists and researchers around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll hear firsthand from an immunotherapy expert who will tell us how cancer treatment today is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can submit your question at any time throughout the webinar by typing in the Q&A box on the screen. Also, you can follow the conversation on Twitter by following the handle @CancerResearch or the hashtag CRI webinar. You can submit your questions also socially, although we prefer if you submit them through the Q&A pod just to make sure that we see them. Now it's my pleasure to introduce today's speaker. Dr. Jedd Wolchok is Chief of the Melanoma and Immunotherapeutics Service, an Associate Director of the Ludwig Center for Cancer Immunotherapy, and the Lloyd J. Virginia and Daniel K. Ludwig Chair in Clinical Investigation at Memorial Sloan Kettering Cancer Center. And he's an associate member of Ludwig Cancer Research. His research focuses on the development of innovative ways to use the immune system to treat cancer. He has spearheaded clinical trials and immunotherapies that have saved lives, in some patients reversing very advanced cancers, sometimes to the point of complete and durable remission. And he's played a vital role in getting ipilimumab, pembrolizumab, and nivolumab to FDA approval. In addition to his appointments at Memorial Sloan Kettering and Ludwig, Dr. Wolchok is also a member of the Cancer Research Institute family. Not only is a funded scientist, but he's an associate director of the Scientific Advisory Council of the Cancer Research Institute, and he is director of the CRI Ludwig Clinical Trials Network. So to no further ado, Dr. Wolchok, we're delighted that you could join us and take time out of your busy schedule to tell us about the future of cancer immunotherapy.
Thank you very much, Brian, and thank you to all the attendees for becoming part of this webinar. I think it's wonderful that CRI has put together this series of webinars. I know the previous ones have been extremely well attended and informative, and I think it's very appropriate that CRI should be doing this given its long-standing primacy in the field of immunotherapy for cancer. As many of you know, the last few years have witnessed a very, very rapid series of progress and events in the development of effective immunotherapies for cancer. This is a field that, although it is not new, the most significant progress has happened just in the past 5 or 10 years. And much of this has actually been work that was funded by CRI. The past few years, we have heard about 2 different areas of immunotherapy that have become front and center in the treatment of cancer. One of those is the development of antibodies or other medicines that either block critical inhibitory steps in the immune system's regulation or agents that, for lack of a better analogy, step on the gas of immune mechanisms. And there are several of these that I'll talk about in greater detail. The other area where there's been significant progress is in what is called cell-based therapy, where usually T cells are engineered or expanded outside the body. T cells are cells that are part of the immune system. They are best known as virus or bacteria-fighting cells. but they also can have a very potent effect on cancer. And we know that T cells can now be engineered and expanded in the laboratory, administered to patients. And some of these approaches have been termed CAR T cells or tumor-infiltrating lymphocyte approaches. And they have shown significant activity, mainly at this point in blood cancers, such as leukemia and lymphoma, but other approaches in solid tumors are now also being evaluated. My area of expertise is in what is called the checkpoint blocking antibodies, and I just want to go into a little bit more detail there. The immune system is a very powerful organ system in the body, and as such, it is regulated by many different positive and negative steps that either get it activated or calm it down. We know it's important to get the immune system activated in order to fight disease. But we also know that if the immune system becomes too activated, it can actually cause disease as well, such as autoimmune disease. And the best known examples of autoimmune disease include rheumatoid arthritis, lupus, multiple sclerosis, inflammatory bowel disease, and psoriasis are some examples. So we see that the immune system can be very important in protecting our bodies from dangerous pathogens and cancer, but also when it is not regulated in a careful manner, it actually can lead to damage. So through careful study in laboratories of what are the molecular switches that turn the immune system on and off, we've become aware at this point of two very important ones. The first is called CTLA4, which was studied in great detail by the head of CRI Scientific Advisory Council, Dr. Jim Allison. And Dr. Allison, along with Dr. Jeff Bluestone, described CTLA4 as a negative regulator of T cells. Dr. Allison went on actually to hypothesize in a very visionary way that if you could block this inhibitory pathway, you might allow T cells to fight cancer better. And through his work, a medicine which is now known as ipilimumab was developed that was actually the first medicine to improve overall survival for patients with metastatic melanoma. And we also know that using ipilimumab resulted in a unique group of adverse events or toxicities, side effects that in some cases were very severe because this mimicked some of the same findings that we see in autoimmune disease. And so oncologists had to become familiar with how to control the side effects of immunotherapy, since they're very different from chemotherapy or targeted therapy. But we have learned to do that. And we now know that ipilimumab can save lives in patients with melanoma. And in addition to that very important accomplishment, we also saw that the success of ipilimumab really led to additional focus and investment on other immune regulating pathways, such as the PD-1 pathway. We've heard a lot about the PD-1 pathway recently, and I want to go into a little bit more detail about what's important about PD-1. PD-1, like CTLA4, is a protein that is found on the immune cells called T cells. It comes up on the surface of these immune cells after the immune system becomes activated, and its role, once it is on the surface of these immune cells, is to mediate the turning off of these immune cells as a way to, again, protect the body from getting overstimulated. PD-1 binds to several different other immune molecules, one of which is called PD-L1. And PD-L1 actually can be expressed on a wide variety of tumor cells. This is a very clear example of how tumor cells have evolved ways to protect themselves from immune attack. Because what happens when PD-L1 that is expressed on tumor cells binds to PD-1 on the T cell is that the T cell dies. And so this is a very clear and understandable mechanism for how a tumor cell faced with immune attack can defend itself. It puts up onto its surface a molecule which leads to the death of the T cell that is actually coming to attack it. And so with that rationale, many different pharmaceutical companies have developed medicines that block either PD-1, the protein on the surface of T cells, or PD-L1, which is present on the surface of tumor cells, as well as on some other immune cells where it is normally found. Two of these medicines, nivolumab and pembrolizumab, have been approved for use in melanoma, and nivolumab has also been approved for use in lung cancer. These are very important accomplishments for our field because these approvals have come just in the past few years. After many, many decades of trying, we are now seeing more and more medicines being developed that use the immune system to treat cancer. And they are being approved in some cases based upon improvement in how long patients are living, not necessarily just for short-term regressions. And this is a major advantage of immunotherapy. The responses that we see are not short-lived in general. They tend to last for months or years. And this is really as a result of the mechanism of action. The immune system is a living, dynamic group of organs. We know that when we are vaccinated against infectious diseases, our immune systems remember those vaccine responses for decades. And so we can see that the immune system has a memory. And what we're hoping to do using immunotherapy is to induce this type of memory response to targets on tumors. So to get back to PD-1, we not only have seen activity of PD-1 or PD-L1 blocking antibodies in melanoma, but we also have seen it in fact, the #1 cause of cancer death in North America, which is non-small cell lung cancer. This is critically important because it shows that immunotherapy can be relevant in very, very common diseases. In addition to lung cancer, where, as I said before, nivolumab has an approval, we are now learning about activity of the PD-1 or PD-L1 blocking antibodies in many other diseases. These include bladder cancer, kidney cancer, head and neck cancer, liver cancer, and importantly, blood cancers as well, because all the other tumor types that we spoke about before are solid tumors. But we now know that PD-1 blocking medicines can have a very high degree of activity in some kinds of lymphomas, most especially in Hodgkin's disease that has not responded to other forms of therapy. So for most of these diseases, the types of responses that we're seeing are durable. They are not found in 100% of patients, but for Hodgkin's disease, 70 or 80% of patients have been seen to benefit. In lung cancer, PD-1 blockade is more in the 20 to 30% range in some studies. What is very important is that we begin to understand why some patients, even within a given diagnostic group, say lung cancer, will have benefit from these medicines while others seem not to. So one very important area of study is looking at biomarkers. What are some properties of patients and the tumors that they have that either leads to sensitivity or resistance to immunotherapy? And we and others have been studying this very carefully. There are some tumor types that seem to attract an immune response at baseline before any therapy is given. So the tumor growing in the patient's body is attracting the attention of the immune system, but the tumor is actively evading the efforts of the immune cells to kill it through mechanisms such as the use of PD-L1 on its surface, as I described before. So again, we'll concentrate on lung cancer for a moment. Within lung cancer, what are some properties that we see in patients who do or do not respond to immunotherapy? Well, one observation that has been made, and this does need more study, is that patients whose lung cancer seemed to have resulted from tobacco exposure appear to be more likely to respond to immunotherapy. Perhaps because the tobacco exposure causes more genetic changes to occur in that tumor and make it look more different than the normal lung cell to the immune system. That might allow for more immune recognition of the tumor at baseline, and therefore a higher likelihood that when we use medicines that accelerate immunity, there are already immune cells surrounding the tumor but being held in check, and we are removing the brakes on those immune cells and allowing them to work better in treating the tumor. So I don't want anyone to walk away from this webinar saying that, oh, I should smoke because it's going to make the immune system work better, or it's going to make immunotherapy work better. That could not be further from the truth. Smoking is still to be avoided, in every instance. But what we are noting is that patients whose lung cancer may have resulted from tobacco exposure may be more likely to respond to immunotherapy. Similarly, we see high activity of immunotherapy relative to other cancers in melanoma, perhaps because the skin cells that give rise to melanoma have been exposed to the putative cancer-causing agent for melanoma, which is ultraviolet light, many hours out of the day, especially now, like in the summertime here in North America. And so there is an accumulation of genetic damage in those melanomas, which may sensitize to the use of immunotherapy. So we can begin to think about ways in which we can identify groups of patients who are more likely to respond. And if we go down the list of diseases that seem to be responding to PD-1, we can see this theme recurring. Head and neck cancer, or larynx cancer, has two different reasons why it may respond to immunotherapy. One is that it can result from tobacco exposure, but it also can result from HPV infection. And one might imagine that if there is a tumor that results from a viral infection, that might be a tumor type that the immune system is interested in because of the immune system's importance in controlling viral infection. We have also seen high activity, again, relative to other cancers for immunotherapy, in bladder cancer, which also is a tobacco-related disease. We've also recently understood that some subtypes of breast cancer, specifically what is called triple-negative breast cancer, and that is a term used to describe breast cancers that don't express the two well-known hormone receptors, the estrogen receptor and the progesterone receptor, and also don't express an oncogene called HER2. That so-called subtype of triple-negative breast cancer seems to be more sensitive to immunotherapy than other subtypes of breast cancer. At the American Society for Clinical Oncology meeting a few weeks ago, we actually learned that liver cancer, which again in some cases is virally mediated, can in fact be responsive to PD-1 blockade. We also learned that some subtypes of colon cancer, which are caused by an accumulation of mutations in some patients who have a familial syndrome called Lynch syndrome, where they can develop colon cancer or uterus cancer that is heavily laden with mutations, can actually respond to immunotherapy. So we are learning every day about more and more types of cancer that can respond to immunotherapy for a variety of reasons. Now, one other area that we and others have been focused on is how to increase the number of patients who have these durable responses to immunotherapy. We spoke about CTLA-4 blockade with ipilimumab. We've spoken about PD-1 pathway blockade. And many investigators in the field, including the group here, has done some work looking at combinations of these approaches together. We recently presented some data using ipilimumab together with a PD-1 blocking medicine called nivolumab, and showed quite a high response rate in patients with melanoma, almost 60%. And that is very exciting for a disease that until very recently has had very few effective treatment options. This combination approach is also being explored in numerous clinical trials, including some that are sponsored by CRI and the Ludwig Institute through its clinical accelerator network, looking at this combination in a variety of different cancers. And we're very excited to see these studies moving forward to try to increase the reach of immunotherapy into other diseases. Now, I think it's important to remember that other forms of anti-cancer therapy, chemotherapy, targeted pathway inhibition, such as BRAF inhibitors, MEK inhibitors, EGFR inhibitors, ALK inhibitors for lung cancer, and radiation therapy continue to play a very important role in treating a broad spectrum of cancers. We believe that immunotherapy could be really included in treatment programs to increase the durability of responses seen with these other types of therapy which target the tumor directly. That's really the difference between immunotherapy and other forms of anti-cancer therapy. Other forms of anti-cancer therapy are largely pointed toward elimination of the cancer cell directly by stopping it from dividing. Immunotherapy is treating the patient, and the patient is treating the tumor. And so these are very different, but potentially, we believe, complementary or even synergistic approaches. And I believe that the next 5 years, to get back to the introduction that Brian provided, is going to be focused on how we rationally and intelligently combine immunotherapy with other forms of anti-cancer therapy to improve the number of patients who respond. We really want to see immunotherapy add to the effect of the many other anti-tumor agents which have been developed so far. We believe that we have some leads in this area. I'm very fortunate to be part of a Stand Up to Cancer Dream Team, actually several of them. One of them, which is co-funded by CRI, looking at immunotherapy, and then another one in lung cancer, which is looking at how to best combine agents that block an oncogene called KRAS in lung cancer with immunotherapy. So I think that the field really is moving toward this approach of combinations, and not just combinations of immunotherapies with other immunotherapies, such as I mentioned before, but immunotherapies with agents that directly target the tumor cell. There are other investigators, some of whom are actually part of CRI's network, that are looking at combining these checkpoint blocking antibodies with cell therapies, with CAR T cells, or with the adoptive T cell therapies to try to enhance activity of both of those approaches. So I believe that we have accomplished quite a lot in the past few years. Multiple new immunotherapies approved based upon very, very hard endpoints, such as patients living longer, which is really the goal. We now seek to improve the number of patients who are benefiting, as well as look more closely at the number of diseases that are showing sensitivity to immunotherapy. So it might be good to open up questions. Brian, what are your thoughts on that?
Yeah, I think that's a great idea, Dr. Wolchok. Thanks for that kind of excellent laying down of the groundwork of where we are today, what's been accomplished in quite the recent past, which is, of course, built on many, many decades of research, and then also kind of looking forward for patients. So we have gotten a number of questions, so I'll go ahead and share them with you. The first one is getting back to side effects. You mentioned briefly earlier on about the side effects associated with immunotherapy, but the question is, how are these side effects different from chemotherapy? And in general, not just the side effects, but in general, what is the treatment experience like for a patient who's receiving immunotherapy compared to a patient being treated with conventional therapies?
That's a really important question, and I thank the person who asked for bringing it up. The experience of receiving the treatment in some ways is quite similar because these the checkpoint blocking antibodies that I spoke about are administered very much in the same way that chemotherapy is administered. Patients come to an outpatient treatment unit. They have an IV infusion, which usually lasts somewhere between 30 and 90 minutes, depending upon the particular medicine being used. And then they go home. Um, that is really where some of the similarity ends. Whereas to compare with chemotherapy, the side effects may become apparent quite quickly. With immunotherapy, the side effects may take weeks, even months, to become apparent. And they're different in nature. Chemotherapy is most infamous, really, for a pattern of side effects that include loss of hair and nausea and vomiting. And the nausea and vomiting, we've done a good job as an oncology community of controlling that. But there are effects on blood counts and the development of mouth sores and things of that nature. Immunotherapy, because of a different mechanism of action, produces inflammation of different body parts. In some cases, this is not very serious. It can be something as simple as itching of the skin, for which we can use some creams. But it can, in some cases, be very serious, and it can involve inflammation of the intestines, which, if it's not treated properly with medicines like prednisone, can actually lead to patients getting very unwell. And in fact, a small number of patients have died from some of these side effects. So they're very different from chemotherapy. They need to be treated differently. But in general, we find the side effects to be reversible for the vast majority of patients. The patients who don't have reversible side effects are those in whom the immune system has, for reasons that we don't completely understand yet, decided to infiltrate some endocrine organs in the body. And I'll name two of them, the thyroid and the pituitary gland. The thyroid, I think we all know, is a gland that sits in the neck that controls our metabolism. The pituitary gland is a very small gland that sits right in the middle of the brain. And it controls, in sort of like a master regulator way, many of the other glands in the body. And if the immune system becomes hyperactivated because of the use of immunotherapy, and these glands stop working because they become full of immune cells, then these glands may no longer produce the hormones that are so important for our body's normal functioning, and some patients may need to take lifelong hormone replacement.
All right, thanks very much for that. You spoke a lot about the checkpoint inhibitors, and understandably, that's where a lot of the excitement is right now. That's where we're seeing a lot of the most dramatic and durable remissions of some previously intractable cancers, and so that's wonderful. But there are, of course, many other, or several other approaches, such as vaccines and adoptive T-cell therapy, CAR T-cells, oncolytic viral therapies. Is there anything you can say about those? When do patients have access to those now, or will they enter the mainstream of cancer treatment in the next 5 years, or if not the next 5 years, when?
That's a great point. So some of these approaches that you mentioned, we did talk about CAR T-cells and adoptive T-cell therapy. Those are entering the later stages of clinical development. They're available through clinical trials. In fact, all of the approaches that you mentioned are available through clinical trials. There's a long history of attempts to vaccinate against cancer with specific injections that try to educate the immune system to recognize certain targets. CRI, through its network with the Ludwig Institute, has been a major supporter of those clinical trials, but there are others as well. I think that those will continue. They very likely will continue in combination with checkpoint blocking antibodies as a way to perhaps steer the immune system toward targets of interest. There are also other immunotherapies that don't block checkpoints, but rather activate positive signaling pathways, so sort of stepping on the gas rather than cutting the brakes. And some of those agents are in clinical trials right now, agents that activate the pathways such as the GITR pathway, the OX40 pathway, another pathway called the CD40 pathway, or another one called the CD137 pathway. Those are all agents that are now currently in clinical trials. You mentioned oncolytic viruses, which are viruses that have a particular specificity to killing tumor cells. And by injecting those viruses into tumors, it may actually attract the attention of the immune system to those tumors. And so we are very interested in this area. There is a product called TVEC, which is a disabled form of herpes virus that has been through phase 3 trials in melanoma and has shown some very significant activity. That is awaiting a decision by the FDA about whether it will be approved, but there was a favorable review by a group called the ODAC, the Oncology Drug Advisory Committee, a few weeks ago about that. So we now believe that the use of oncolytic viruses may become also an important part of anti-cancer therapy, either alone or in combination with checkpoint blocking antibodies. There was data at the ASCO meetings about the use of TVEC along with ipilimumab. There are clinical trials launching, looking at anti-PD-1 with TVEC. And so I think this idea of using viruses in combination with checkpoint blockade has much scientific support.
So let's stick with melanoma. I know that's an area that you have particular expertise in. We have, for instance, a patient who was treated with the ipi-nivo combo, responded well, but then after about 11 months began to progress again and is now on, I think, MEK inhibitors, and a few other targeted therapies. And his question is, now there's nivolumab or pembrolizumab, I'm sorry, and should he consider pembrolizumab? Is there a different mechanism of action between that and nivolumab?
It's a great question. Sorry to hear that the patient has had a recurrence, but very fortunately there are, as you can see, multiple different lines of therapy which can provide benefit, unlike a few years ago. It's a very important question. We have two medicines, nivolumab and pembrolizumab, both PD-1 blocking antibodies, both approved to treat melanoma, both showing very similar percentages of patients who have response, and both showing very similar side effect profiles. But we don't know whether one of them can substitute for the other in the context of recurrence. So there is no data to speak of. And I think it's a very open question at the moment. One area that we should review, though, is that for patients, if we turn back the clock maybe about 5 years to a study that was published by my colleague and another member of the CRI Clinical Trials Network, Dr. Steve Hody from Dana-Farber. Dr. Hody's landmark publication in the New England Journal of Medicine showed, in fact, that patients who responded to ipilimumab by itself and then had a recurrence and were then retreated with ipilimumab had a significant likelihood of returning to a state of disease control or having a favorable effect. Although that's not part of the current label for use of ipilimumab, we do know that there is data from that phase 3 trial to support that.
All right, thanks. And another question from not a patient, but a caregiver of a patient whose daughter has non-squamous cell non-small cell lung cancer. The approval of Opdivo was in squamous cell non-small cell lung cancer. So are there any immunotherapies in development or nearing approval in that indication?
So there was actually data at the ASCO meeting for nivolumab in patients with non-squamous, non-small cell lung cancer, showing activity in that subtype as well. And I believe that many of the other PD-1 blocking agents, pembrolizumab, the PD-L1 blocking agents from both MedImmune and also Genentech, NPDL-3280A, as well as MEDI4736, also have activity across subtypes of lung cancer, non-small cell lung cancer.
That's great. And now what about ovarian cancer? This is, ovarian is one of those types of cancers that often is not detected until it's already quite advanced. And the typical remission period at 5 years ends and many patients recur. Up to, I think, 80%, maybe even more of patients diagnosed with stage 4 recur. What developments have there been in ovarian cancer immunotherapy?
It's a great point, and as you know, one of the leaders of the CRI and Ludwig Clinical Trial Network, Dr. Kunle Odunsi from Roswell Park, is a major figure in this field, and he and others, Dr. George Kukos, who's at Lausanne at the Ludwig, and Dr. Paul Sabatini here, have done very important work looking at different immunologic approaches to anti-cancer therapy of ovary cancer. And what we now know, in fact, is that the use of checkpoint blocking antibodies, such as nivolumab, can, in fact, benefit some patients with ovary cancer. A small study done in Japan, presented at ASCO last year in 2014, which showed about a 20% response rate in ovary cancer. And in fact, the clinical trials, which I mentioned before, looking at combinations of CTLA-4 and PD-1 blockade, in both the CRI Ludwig-sponsored trials as well as others, have included patients with ovary cancer to try and address this important question.
Here's a question that I have not come across in quite a while, and it's a question about hyperthermia and combining hyperthermia where you raise the body temperature of a patient while administering something in order to boost the immune system. And I know Cancer Research Institute funded some research, I believe in Germany, about 20 years ago on hyperthermia. I'm wondering, can you just speak briefly about that? What's the status of that?
So I wish I could, but I really don't have a lot of knowledge in that area. I know that hyperthermia is used in conjunction with some chemotherapy regimens to try to increase the likelihood of benefit, but I'm really not facile enough with that field to comment intelligently.
All right, fair enough. What do you know about combining checkpoint inhibitors with ALK inhibitors in non-small cell lung cancer? Is there any data showing synergistic activity there?
So I think this is an important area of combinations of targeted therapies with immunotherapies, and the data is still really being generated looking at these. We have to look at the important basic questions about the schedule of how to combine these approaches together, making sure that one approach doesn't get in the way of the other. But I think that's a very intelligent question, and it's an area of intense research focus right now in the clinic.
And do you know anything of anti-PS targeting antibodies and whether or not they may work, like such as bevacizumab, and whether there's any synergy there with checkpoint inhibitors?
So the anti-PS or phosphatidylserine antibodies, we are involved in a collaboration to look at that. It's not bevacizumab, it's a different antibody, bevacizumab, and I think that is an area of deep interest to my group and others to try to establish what are the role of anti-PS antibodies in immunotherapy regimens.
All right, thanks. And I think the last question that we'll have for the day is about colon cancer, and there's been limited success of PD-1 therapy in colon cancer. What's the future of immunotherapeutics in this particular indication?
So I think that's a great question, and again, I think we have to begin to think about diseases by more specific subtypes. So I don't think it's sufficient to talk about colon cancer alone. We have to talk about colon cancer that is related to syndromes like Lynch syndrome, or what's called MSI high or microsatellite instability high lung cancer, which has colon cancer, which has many mutations. The group at Johns Hopkins presented very compelling data about this at the ASCO meeting. And so we now know that it's important to look not just at what the shape of the cells is under the microscope and what organ they came from, but we also need to understand what genetic errors are present in those tumor types, which may inform us about whether or not immunotherapy can work. There may have been some general thoughts about lack of activity of immunotherapy in colon cancer, but that was based upon large populations, unselected populations of patients. Now we understand that if you focus on specific subtypes of colon cancer, which may be particularly sensitive to immunotherapy, such as this Lynch syndrome kind of colon cancer, we may see high activity of anti-PD-1.
So this is definitely a trend that I hear a lot at scientific conferences, and looking at the research proposals that come through to the Cancer Research Institute when it comes to subtyping tumors and developing therapeutic approaches that are specific to that. And that, I think, is going to be an increasing trend as the technology progresses and as our knowledge of how these individualized profiles of different tumor types based on each specific patient's tumor are going to affect treatment.
Right.
So, well, thank you again, Dr. Wolchok, for that. I'd like to thank again our sponsor, Amgen, for providing support for this, the final installment in our series of webinars taking place during June, which is Cancer Immunotherapy Month. So please, if you'd like, be sure to check out some of the other webinars that we've conducted throughout the month of June, as well as in the past. You can see them at cancerresearch.org/webinars. And if you'd like to learn more about immunotherapy for various forms of cancer, presently we have 10 forms of cancer that are represented on our website, theanswertocancer.org. You can learn about what immunotherapies are available, and you can also be connected to a clinical trial testing an immunotherapy in your type of cancer. So go to the answertocancer.org patient portal for patients and caregivers. And if you'd like to learn more, of course, about the Cancer Research Institute, our history, our mission, and what we're doing today to be the leader in immunotherapy, and if you'd like to get behind our mission and make a donation to support our important work, you can do that by visiting cancerresearch.org. I'm Brian Brewer with Cancer Research Institute, thanking you.